Hello and welcome to Mull by All Review. I'm your host, Ryder, and this week we're going to be looking at two papers. The first is the Inflammasome Pyrin contributes to Pertussis toxin-induced IL-1 beta synthesis, Neutrophil intravascular crawling, and Autoimmune Encephalomyelitis, from the laboratory of Dr. Luc Valliere and first authored by Alain Dumas. As you're all aware, EAE is the mouse model of multiple sclerosis, and PTX, pertussis toxin, is an adjuvant commonly used to increase an immune response. Figure 1. The qPCR on IL-6 and IL-1 beta. IL-6 and IL-1 beta are both increased in leukocytes in the peritoneum after PTX injection, but not after PBS. 1B. 1B, ELISA. Only IL-6 is increased in the circulation, but IL-1 beta and IL-6 are both increased in the peritoneum. Figure 2, bone marrow chimeras. Things that depend on IL-6 in the recipient. Peritoneal IL-6, plasma IL-6. Things that depend on IL-6 in the donor, nothing. Now IL-1 beta. Things that depend on IL-1 beta in the recipient, none. Things that depend on IL-1 beta in the donor, IL-6 and IL-1 beta. Conclusion: IL-6 is required in radio-resistant cells. IL-1 beta is required in hematopoietic cells. Lack of IL-6 does not affect IL-1 beta. Therefore, IL-6 is downstream of IL-1 beta. Figure 2b. Depletion of CD11b positive myeloid cells blocks IL-1 beta. Thus, myeloid cells are upstream of IL-1 beta. 2c, by cell sorting, macrophages upregulate IL-1 beta mRNA, neutrophils upregulate IL-1 beta mRNA. 2e, 2f, IL-6 mRNA co-localizes with stromal cells. Stromal cell cultures respond to IL-1 beta by producing IL-6. Figure 3. Does this IL-6, IL-1 beta signaling go through the inflammasome? IL-1 beta and IL-6 are suppressed in TLR4 KO mouse, caspase-1 KO mouse, and Pyrin KO mouse, but not NLRP3 KO mouse. PTX toxin is an enzyme. Does the effect depend on the enzymatic activity? Figure 4, A. Injection of just PTX A or B subunit separately does not cause any of these cytokine production. Likewise, enzymatically dead PTX does not cause any increase of IL-1 beta or IL-6. And by Western blot, in cell isolates, only when the full enzymatically active PTX is injected do you see IL-1 beta, cleaved IL-1 beta, caspase-1, cleaved caspase-1, TLL4, and MyD88, the signaling molecule of TLR4. Side note, PTX is an ADP ribosyl transferase, often used by bacteria as a pathogenic enzyme to deactivate host proteins. Figure 5. Pyrin mRNA is upregulated and it's dependent on TLR4 expression. Pyrin and IL-1 beta correlate with each other in their expression levels. Neutrophils are the main producers of Pyrin mRNA, and Pyrin protein is detectable after PTX injection. Figure 6. Impact of Pyrin and PTX on EAE. Pyrin is required for the infiltration of neutrophils into the brain. but not for the infiltration of T cells. Pyrin KO mice have higher body weight, they have a later onset of disease, and their disease is a little bit milder. Last figure. Finally, Pyrin is not required for the development of passive EAE induced by adoptive transfer of CD4 T cells. In summary, microbial agents can aggravate inflammatory diseases such as MS and its animal model, EAE. An example is Pertussis toxin, a bacterial virulence factor commonly used as an adjuvant, but whose mechanism of action is unclear. We have reported that PTX triggers an IL-6 mediated signaling cascade that increases the number of leukocytes that patrol the vasculature by crawling on its luminal surface. In the present study, we examined this response in mice lacking either TLR4 or Inflammasome components and using enzymatically active and inactive forms of PTX. Our results indicate that PTX, through its ADP ribosyl transferase activity, induces two series of events upstream of IL-6. First, the activation of TLR4 signaling in myeloid cells, leading to pro-IL-1 beta synthesis. And two, the formation of a Pyrin-dependent inflammasome that cleaves pro-IL-1 beta into its active form. In turn, IL-1 beta stimulates stromal cells to secrete IL-6, which is known to induce vascular changes required for leukocyte adhesion. Without Pyrin, PTX does not induce neutrophil adhesion to cerebral capillaries and is less effective at inducing EAE in transgenic mice with encephalomyelitis T lymphocytes. This study identifies the first microbial molecule that activates Pyrin, a mechanism by which infections may influence MS and a potential therapeutic target for immune disorders. Now, for our second paper here today, loss of CD24 in mice leads to metabolic dysfunction and a reduction in white adipocyte tissue. This is a paper that comes from Nicholas Fairbridge in the laboratory of Dr. Sherry Christian at the Memorial University of Newfoundland. Okay, so just a bit of background. White adipose tissue, or WAT, it's fat. It's also an endocrine organ that secretes hormones such as adiponectin and leptin. There are a few differences between subcutaneous WAT and visceral WAT. Subcutaneous WAT contains more pre-adipocytes and fewer differentiated adipocytes. Finally, pre-adipocytes express CD24 while adipocytes do not. The reason for this downregulation is unknown. Figure 1, the CD24 KO mice. In all these figures, dashed line is black 6, solid line is CD24 KO. CD24 KO mice have less fat and a concomitant increase in lean mass and bone mass. If you dissect out the subcutaneous or visceral WAT from these mice, you'll find the same result. CD24 KO mice have smaller white adipose tissue. Figure 2, can CD24 null mice still respond to a lipogenic diet? In response to a high sucrose diet or a high fat diet, CD24 KO mice will still gain weight, indicating that CD24 null does not block the recognition of triglycerides from either VLDL or chylomicrons. Table 1, if you look at serology from these KO animals, glucose is up, insulin is down, adiponectin and leptin are down, and free fatty acids are up. Alright, and if you feed mice on a high fat or high sucrose diet, you can still see table 2, glucose is still higher than normal, and adiponectin is still lower than normal. So I guess this might be due to the lower mass of white adipose tissue in these CD24 KO animals. Figure 3 is the KO mice response to glucose and insulin. I'll just show the summary here. So it turns out that the KO mice are actually less responsive to glucose, especially evident in the high-fat diet, but under normal conditions, they're more responsive to insulin. Okay, finally, figure 4. So we know that CD24 knockout mice have alterations in their hormone levels, their fat content, and their response to glucose and insulin. But there's one question that remains: is their white adipose tissue hypoplastic or hypotrophic? They measured the cell area and cell number of WAT from wild type and mutant mice, and they did not observe any difference in the cell number. Conclusion: the WAT from CD24 KO mice is hypotrophic. To summarize, CD24 is a glycophosphatidylinosytol-linked cell surface receptor that is involved in regulating the survival of or differentiation of several different cell types. CD24 has been used to identify pre-adipocytes that are able to reconstitute WAT in vivo. Moreover, we recently found that the dynamic upregulation of CD24 in vitro during early phases of adipogenesis is necessary for mature adipocyte development. To determine the role of CD24 in adipocyte development in vivo, we evaluated the development of the inguinal and interscapular subcutaneous WAT and the epididymal visceral WAT in mice with a homozygous deletion of CD24. We observed a significant decrease in WAT mass of 40 to 74% in WAT mass from both visceral and subcutaneous depots in male mice, with no significant effect in female mice compared to wild-type sex and age-matched controls. We also found that CD24 KO mice had increased fasting glucose and FFA, decreased fasting insulin and plasma leptin. No major differences were observed in the sensitivity to insulin or glucose on the whole, or in circulating triglycerides, total cholesterol, HDL, or LDL cholesterol levels between wild-type and null mice. Challenging the CD24 KO mice with either high sucrose or high-fat diets that promote increased adiposity, increased WAT mass, and fasting insulin, adiponectin, and leptin levels, as well as reducing the sensitivity to insulin and glucose to the levels of wild-type mice in the same diet. The CD24-mediated reduction in fat pad size was due to a reduction in adipocyte cell size, hypertrophy, in all depots with no significant reduction in pre-adipocyte or adipocyte cell number. Thus, we have clearly demonstrated that the global absence of CD24 affects adipocyte cell size in vivo in a sex and diet-dependent manner, as well as causing metabolic disturbances in glucose homeostasis and FFA levels. Well, I hope you enjoyed my commentary on this week's edition of Mole by All Review. If I made any mistakes, please let me know in the comments. The links to all articles will be in the description. Thanks for watching!